Hello everyone, welcome to the lecture covering the physiology and mechanisms of the atherogenic lipid triad. In this presentation, we will cover the following topics. First, we'll define what the atherogenic lipid triad is. Then we'll discuss the structures that make up a lipoprotein and how they are classified into groups. Next, we'll discuss what the basic blood lipid metrics tell us about these lipoproteins. Then we will move into the mechanisms of normal lipid enzyme activity and when that activity becomes abnormal and pathogenic. This will create the story that explains this atherogenic lipid triad. This lecture will conclude with a mention of a few advanced blood lipid metrics that one can ask for at their doctor's office. So here we go. The atherogenic lipid triad is a phrase that has been coined by cardiologists, lipidologists, and endocrinologists that groups three interconnected blood lipid metrics. Together, the three components that make up this triad have been found to significantly correlate to increased risk of developing atherosclerotic cardiovascular disease. The correlation of this triad to disease risk is much greater than the correlation of LDL alone or any of these components alone. What's interesting is that these three components are physiologically connected, meaning they, more often than not, present themselves in blood work altogether. We'll discuss this connection later in the lecture. So, what are these three components? One is high triglycerides, also known as hypertriglyceridemia. Two is low HDL-C. This is the metric we would rather see high. And three is a high number of small LDL lipoproteins. There are genetic polymorphisms some humans possess that predispose them to develop these negative metrics. We will not cover the genetic differences in this lecture, primarily because it's a whole other discussion in itself, and these polymorphisms are only present in a tiny percentage of individuals. The majority of cases in the U.S. develop due to lifestyle and behavior influences. On the last slide, I mentioned the term lipoprotein. Let's define what this is. Lipoproteins are a spherical structure produced by your intestines or your liver and carry lipids or fats throughout your bloodstream. Lipids are hydrophobic, meaning they are repelled by water and therefore cannot float all by themselves through our blood. 55% of your blood is plasma, and mainly plasma is water. So in order for lipids to move about the body through the bloodstream, they have to be packaged in these particles called lipoproteins. I want you to picture a lipoprotein like a vehicle that is shaped like a basketball. The inside this vehicle are our various lipids that it's carrying. On the outside surface of this spherical vehicle, phospholipids line themselves up in their hydrophilic manner to create the outer barrier. Tucked here and there between the phospholipids, there are occasional molecules of free cholesterol to provide more structure to the surface. This is illustrated on this slide in the left image. The cargo of this vehicle, the lipids, are held inside the core of the particle. Inside you'll find molecules of triglyceride, as well as molecules of cholesterol ester. Cholesterol esters are basically free cholesterol molecules that have been esterified or converted to their storage form. Additionally, embedded in the surface membrane of the particle, you will find various other protein structures called apolipoproteins. These apolipoproteins, or also called apos, give the lipoprotein its signature, as well as serve as a ligand with receptors on cells throughout the body. These apos identify what the particle is and allow the particle to bind to receptors to then deliver or take on their lipid cargo. So why do we need to move all these lipids around the body? Well, each lipid has a very important role. Triglycerides are used in every cell that contains a mitochondria to produce ATP. Triglycerides provide energy. Cholesterol esters, or free cholesterol, are used by every cell for membrane structure. They are used to produce steroid hormones. They are used to produce bioconstituents, as well as facilitate vitamin D absorption from our food. And if you remember from basic biology class, phospholipids are the primary molecule that make up every cell membrane in our body. Lipids are very important. We need all of them for proper cell function, and lipoproteins are what deliver these lipids to the places in the body that need them. The proportion of each lipid within the core of the particle varies depending on the classification of the particle. Some have more triglyceride than cholesterol ester, and some have more cholesterol ester than triglyceride. Cholesterol ester is a denser molecule than triglyceride, so lipoproteins that contain more cholesterol ester than triglyceride will be considered denser. This density difference due to the cargo differences is one way these lipoproteins are classified into light groups. Additionally, the apolipoprotein on the particle's surface varies between the particle classifications. This is another way lipoproteins are distinguished from each other. Let me introduce you to all the lipoprotein classifications, starting with the lowest density particle, the chylomicron. The chylomicron is produced in the enterocyte, our intestinal cell. Primary purpose of this lipoprotein is to deliver postprandial triglycerides to our cells. It takes it from the food that we eat and delivers it about the body. The next type of particle is smaller and contains less triglyceride. It's more dense than the chylomicron, but less dense than the rest of the particles. This is the very low density lipoprotein, or VLDL. VLDLs are produced in the liver. When chylomicrons are done with their job, they return to the liver and the leftover contents get recycled. The liver will take the leftover triglyceride and any other stored or endogenously produced triglyceride and packages them into a VLDL. Then they get released into the bloodstream to keep delivering triglycerides to the cells. As chylomicrons and VLDLs deliver their triglyceride, they get smaller and smaller, and at a point, they are then referred to as remnants. Chylomicron remnants return back to the liver. VLDL remnants can do the same, but VLDL remnants can also keep losing their triglyceride to the point that they become a different particle called an intermediate density lipoprotein, or IDL. Next on the slide, you see the low density lipoprotein, or LDL. LDLs are produced in the liver, but they can also be the end product of an IDL. At this point, the amount of triglyceride in the particle has reduced so much that the majority of the cargo in it is cholesterol ester. So the primary job of the LDL is to deliver cholesterol to the cells that need it. Now, LDLs have a few subclasses within them. Simply put, they are considered large or small. However, some more sensitive lipid tests can identify up to 5 subclasses. More commonly, though, they are just referred to as large LDL or small LDL, with the cutoff division point being approximately the size of the space between each endothelial cell in the blood vessels. Remember this because we'll come back to this later. What about the apolipoproteins now? According to the apolipoprotein classification, chylomicrons, VLDLs, IDLs, and LDLs are collectively considered apolipoprotein B lipoproteins. Each and every one of these particles have one and only one ApoB on them. ApoB48 is the type on the chylomicron, and ApoB100 is the type found on the VLDL, IDL, and the LDL. There is also an obscure lipoprotein within the LDL family that has an apolipoprotein on it called lipoprotein a, or Lp(a). One's presence of these lipoproteins is mostly determined by genetics, but in all cases, you want very little, if any, of these lipoproteins. They are significantly more atherogenic. And last, we have the high-density lipoprotein, or HDL. HDLs are produced by the liver, and they actually have several roles in the body. The most familiar one is that they scavenge excess cholesterol from the endothelial macrophages, but they also serve antithrombotic, anti-inflammatory, and antioxidant roles. According to the apolipoprotein classification, HDLs have the apolipoprotein A1 on them, aka ApoA1. HDLs, however, can have more than one APO1 on them, unlike the ApoB particles, which only contain one each. Take a look at the lower image on the slide. This is a visual of each lipoprotein and its proportion of triglyceride and cholesterol ester inside itself. Remember that cholesterol ester is denser than triglyceride. So if a particle has a greater proportion of cholesterol ester to triglyceride, it will be a denser particle. This is why the terms very low density, and high density end up in the names of these lipoproteins. HDL have the most cholesterol ester to triglyceride in their core, and therefore have the highest density. Chylomicrons have way more triglyceride than cholesterol ester in their core, and therefore are the least dense of every lipoprotein. When you hear the word density, I want you to think of something feeling firmer or feeling more squishy. If you could hold a particle in your hand, the higher the density, the firmer it would feel. The lower the density, the squishier it would feel. For example, a golf ball is denser than a racquetball. It is also smaller and weighs more. If the golf ball were a lipoprotein, it would be the one that contains more cholesterol ester than triglyceride. Take a moment now to read all of the columns in the table image on the slide. Feel free to pause the video for a moment while you read. Another characteristic of lipoproteins I'd like to point out is the plasma residence time. Basically what this means is, once the lipoprotein has been produced, how long does this float around in our bloodstream doing its job? This also varies between the lipoprotein classifications. Take a look at the table. The chylomicron is produced in the intestines after we eat a meal. These particles move throughout the bloodstream, delivering the triglyceride for an hour or less. If you were to measure someone's blood for chylomicrons, you'd have to measure them within an hour after eating, or you wouldn't find very many kilos. VLDLs will also spike postprandially, but remain in the bloodstream for longer, about 4 to 6 hours long. IDLs only last for an hour and are such a small blip of time, you almost never even hear about them in conversation about cholesterol. LDLs and HDLs, however, last in the bloodstream for several days. LDLs about 2 to 4 days, and HDLs about 4 to 6 days. The body is cyclically producing and clearing these two lipoproteins, so there's always a supply in the bloodstream. We now know what the contents and density of our lipoproteins are, and we know how long they float around in the bloodstream. But how many of each do we have? To know total numbers, it takes a special test. But what we can see here are relative numbers. This can give you an idea of how the number of each particle compares to the number of the others. Now this picture on the slide is the relative number for a normolipid individual. When triglycerides increase to abnormal levels, this relative number gets more exponential. For every 1 chylomicron, a normolipid individual will possess approximately 10 VLDL particles. And for every VLDL particle, a normolipid individual will possess 9 to 10 LDL particles. So for each chylomicron, we'd normally have close to 100 LDLs. Picture this. If we were to put all of our ApoB particles in a jar, approximately 98% of them would be LDLs. This gives you an idea of the proportions each particle makes up within our bloodstream at a given time. Our lipoproteins have a job to do. They have lipid to deliver and a time frame to deliver it in. Mostly, they are delivering their cargo to the various cells in the body. However, each lipoprotein can also interact with other lipoproteins and exchange contents. The systemic needs of the body are what drive this lipid trafficking. In order for this to happen, though, enzymes must be present to facilitate these exchanges. Let me introduce to you the enzymes involved. I'll start with lipoprotein lipase. This enzyme is present on numerous cells and hydrolyzes triglyceride from the chylo, VLDL, and LDL particles. This is the enzyme that removes the triglycerides so that they can be used for ATP production in the cell. We like this enzyme. Next we have hepatic lipase. This enzyme is only present on a liver cell and does the similar thing as LPL, lipoprotein lipase. It hydrolyzes the triglyceride from the LDL and the HDL particles at the liver. Third, we have lecithin cholesterol acyltransferase, or LCAT for short. LCAT is primarily responsible for lipidating an HDL particle with cholesterol esters. This means that LCAT removes the cholesterol esters from chylos, VLDLs, and LDLs and puts it into an HDL. This facilitates removal of unneeded cholesterol esters. Last, we have cholesterol ester transfer protein, or CETP for short. This enzyme performs trades. It exchanges one triglyceride for one cholesterol ester between two lipoproteins. Take a look at the slide and see which ones do the trading. CETP can take triglyceride out of an LDL and put it into an HDL in exchange for a cholesterol ester. And CETP can also take triglyceride out of a VLDL in exchange for a cholesterol ester from both LDL and HDL. When and how much it trades depends on the needs of the body. If you were to visit the doctor and have a basic blood lipid panel done, the same panel that can be administered in the HAP lab, you would be able to see the following metrics. Total cholesterol. Total cholesterol in a given amount of blood, this metric tells you that the cholesterol content within all lipoproteins combined. Optimal range is less than 200 milligrams per deciliter. You will also be able to see your LDL-C, or the cholesterol content only within the LDL particles. Optimal range is less than 100 milligrams per deciliter. Next, you will see your HDL-C, or the cholesterol content only within the HDL particles. Optimal range is greater than 40 milligrams per deciliter. You can also see your non-HDL-C, and this is the cholesterol content within the LDL plus the VLDLs and IDL particles. Triglyceride is another number you can see, and this is telling you what the triglyceride content is within all the particles. But remember, VLDLs have a greatest proportion of triglyceride compared to other particles and are the most numerous of that great proportion. Chylomicrons don't really exist in the blood for very long. So primarily, this metric is measuring VLDL content of triglyceride. Optimal range is less than 150 milligrams per deciliter. The lower the better. Last, this panel will also calculate your total cholesterol to HDL ratio. You simply divide total cholesterol by HDL-C and get a decimal. The lower this number is, the better. Epidemiological studies have determined these optimal ranges based on strong correlations between non-optimal levels and cardiovascular disease risk. For example, as LDL-C, non-HDL, total cholesterol, and triglyceride increases, cardiovascular disease risk also increases. And as HDL-C decreases, again, cardiovascular disease risk increases. There is a limitation in these metrics though. You see, the actual lipoprotein particles that are carrying these above contents have a large amount of variability in their proportion of the contents. For example, in the LDL particles, you have a given amount of cholesterol that is measured in this test, right? Well, that amount of cholesterol could be carried by just a few gigantic LDL particles, or it could be carried by a ton of tiny little particles. But this basic test would give you the same LDL-C number despite the particle count. The particle is the truck, the vehicle, and the cholesterol or the triglyceride is the cargo. The same amount of cargo could be carried by tons of little trucks or just one giant truck. The above tests only measure the cargo, not the trucks. Let's talk about why knowing the amount of trucks you have to carry your cargo or your particle count matters. When we're associating lipids to cardiovascular disease, the type of cardiovascular disease we're referring to is atherosclerosis. I'm not going to go into all the process of developing atherosclerosis because I believe you've talked about that already. But I do want to focus in on basically step 1 of this process. Researchers and cardiologists have 2 different stances on this step 1, and they seem to be simply coined as the retention hypothesis and the injury hypothesis. The retention hypothesis states that the endothelium of the vessel leaks lipoproteins into and out of its intracellular spaces all the time, whether or not the endothelium is normal or damaged. Only if the infiltrated material is retained does the pathogenic cascade begin. The injury hypothesis states the endothelium is injured or damaged first, creating increased permeability, allowing the lipoproteins to leak into the intracellular spaces where they otherwise would not be present. This entry provokes a cascade of pathogenic events, initiating the disease process. There are good evidence-based arguments for both of these hypotheses, but at the bottom line, they point to the same basic role the lipoprotein plays. What they both state is that lipoprotein entry and retention is necessary as well as sufficient to provoke the atherogenic cascade. So it doesn't matter which hypothesis is correct. What matters is the particle, the lipoprotein, and its entry and retention. You see, entry of the particle into the endothelial spaces is driven by a concentration gradient. The greater the concentration of particles within the lumen of the vessel, the greater the driving force of pushing those particles into those open spaces. Now, in addition to this concentration gradient, size matters. Which subclass of lipoproteins are likely to fit better into those intercellular spaces? If you remember from the lipoprotein classification slide, it is small LDLs and HDLs. The thing that differentiates those two particles are the apolipoproteins. The ApoB on the LDL gets obscured and funky when the LDL particle becomes as small as the HDL. This deformity in the ApoB makes it very susceptible to binding to the proteoglycans in the vessel wall, therefore increasing its retention capabilities. This deformed ApoB also makes this lipoprotein more susceptible to oxidation, which occurs in the following steps of the atherogenic cascade. So, boom, small LDL particles and a larger number of those small LDL particles are more atherogenic. How in the world do LDLs become small then? And how would we know this about our LDL particles? And how would we know what our particle count is of these small LDLs? If you remember, the basic blood lipid panel that people can get from their regular doctor's checkup does not tell one what the size and count of their lipoproteins are. Those basic tests measure cargo, not the truck. If we were to clearly understand our atherogenic potential, we need to measure the truck. We need to know how big these trucks are and how many we have. I will introduce you to these tests later, but let's first talk about the mechanism of how these LDLs become small. I want to revisit the components of the atherogenic lipid triad. High amount of triglycerides, low amount of HDL-C, and high amount of small LDL particles. As you can see, this increased number of small LDLs is one of the components. All of these components are interconnected though, and it all begins with insulin resistance. The development of insulin resistance can look differently in different ethnicities, and certain population groups are more predisposed to this development than others. For example, people of African, East Asian, and Native American descent have a significantly greater risk of developing insulin resistance than Caucasian descent. Even though there are genetic polymorphisms that expedite this process, the process itself is quite similar. It begins with a mismatch in consumption and utilization. Certain foods and drinks elicit a rapid and large glucose dump from the intestines into the bloodstream. Simultaneously, the pancreas releases insulin to facilitate the glucose from the bloodstream into the cells. Physical activity and movement in general also can facilitate this glucose from the bloodstream into the cells. But if one is sedentary and consuming foods that elicit this high insulin response, the mismatch in consumption and utilization occurs, and hyperinsulinemia results. Hyperinsulinemia does two things. One, it reduces GLUT4 expression on the cells. Now, insulin is actually the key that mobilizes these transporters. But too much insulin is a bad thing. It does the opposite. This is the state of hyperinsulinemia. So this reduction in the transporter means that the glucose remains in the bloodstream rather than being facilitated into the cells. So to achieve glucose homeostasis, the glucose gets converted to glycerol, bound to fatty acids, and becomes a triglyceride to be stored away. The second thing that hyperinsulinemia does is it inhibits an enzyme called hormone-sensitive lipase. If this enzyme gets turned off, which is what happens from insulin, triglycerides are moved into storage rather than being used. Visceral adiposity results from this. These visceral adipocytes have a mind of their own, though. They used to be thought of as inert, but they secretly do bad things. These adipocytes release inflammatory markers into the bloodstream, and they also reduce their release of adiponectin, which is something you want. If the body is chronically in this state of high blood glucose, high blood insulin, high inflammatory markers, and low adiponectin, eventually the glucose receptors on the cell become damaged permanently. Insulin resistance results from this. Once this happens, things start to get wonky with our metabolism. Interestingly, though, insulin resistance has been found to be the pathophysiological mechanism of several diseases. Obviously for type 2 diabetes, but also for metabolic syndrome, Alzheimer's, and heart disease. Insulin resistance can develop in various cells in the body. But when insulin resistance occurs in visceral adipocytes, more wonky things happen as a result. If you remember, insulin normally inhibits hormone-sensitive lipase. This means that insulin turns off this enzyme, and the job of this enzyme is to hydrolyze the triglycerides into free fatty acids so they can be used to make ATP. Insulin turns off this activity and promotes triglycerides into storage rather than utilization. But if insulin resistance develops in these visceral adipocytes, this enzyme is no longer regulated by insulin. So why is this bad? Because wouldn't you want to increase hydrolyzing triglycerides? Well, you'd only want to do this if there was a need for them to fuel ATP, but this is not the case. This unregulated hormone-sensitive lipase in the visceral adipocytes hydrolyzes a ton of triglyceride and releases a ton of fatty acids into the systemic and portal circulatory systems. The result is way too much fatty acids in the blood. So our liver tries to save the day. The liver comes to the rescue and takes up this excess fatty acid from the blood. As the liver takes up this excess fatty acid, it has to adjust its metabolism to this new influx of substrate. There are 4 fates of this excess fatty acid in the liver now. First, the fatty acids can be used by the mitochondria in the liver to produce ATP. This is a very good use of them. However, this process is actually really efficient, which means that very little is actually going to be needed for this process. This does not put a huge dent in that surplus of fatty acid. Second, these fatty acids can fuel the process of gluconeogenesis in the liver. However, blood glucose would need to be low for this process to be stimulated. Obviously, this is not the case here, and likely this fate will not even happen. Third, the liver can pack these excess fatty acids into more VLDL particles, then release these particles into the bloodstream, hoping that somewhere else in the body can use them for energy. The result of this is high VLDL particles in the blood and hypertriglyceridemia. And fourth, the excess fatty acids can be converted back to triglycerides and deposited into the hepatic tissue. If this happens, hepatic steatosis can result, and boom, we have fatty liver disease. Fate 1 uses very little of this excess fatty acid. Fate 2 is likely not even to happen in this circumstance. So what do we have left? Fate 3 and Fate 4. And here we end up with the first component in the atherogenic lipid triad, high triglycerides or hypertriglyceridemia. Lipoprotein lipase, if you remember from the slide on the enzymes, is responsible for hydrolyzing triglyceride from all lipoproteins. So you'd think that this enzyme might be able to help reduce this hypertriglyceridemia that is occurring in the VLDLs. But unfortunately, in this state of insulin resistance, LPL activity also gets wonky. Lipoprotein lipase activity decreases and no longer can hydrolyze triglycerides from the lipoproteins. This happens as a result of 2 metabolic alterations. This is a time where the genetics of an individual can play a part. In certain circumstances, the liver can overproduce another apolipoprotein called ApoC3 and load them onto LDL particles. When these particles come in contact with LPL, the ApoC3 inhibits the enzyme. So, overexpression of ApoC3 turns LPL off. The next mechanism is caused by insulin resistance. The enzyme LPL requires dimerization to become active. Dimerization is basically the bonding of 2 molecules. Insulin resistance increases the expression of a protein called angiopoietin-like protein 4. This protein prevents the dimerization and renders the LPL in its inactive monomer state. This decrease in LPL activity means that VLDLs do not convert to IDLs, then LDLs, because their triglycerides never get hydrolyzed. They basically just stay as VLDLs. In a basic blood lipid panel, this would result in, once again, high triglyceride, but additionally, normal or low LDL-C. LDL has been the focus of action for cardiovascular risk reduction by most healthcare providers. But this all too common circumstance can easily be dismissed because of the normal or even low LDL-C number. Let's revisit two of the enzymes on this slide. Take a look at CETP, cholesterol ester transfer protein. CETP exchanges lipids between two lipoproteins. It basically makes a trade in a 1-to-1 ratio of cholesterol ester with triglyceride. Between the HDL and an LDL, CETP will remove the cholesterol ester from the HDL and give it to the LDL, and it will remove the triglyceride from the LDL and give it to the HDL. Between an LDL and VLDL, it will remove a cholesterol ester from the LDL and give it to the VLDL, and remove a triglyceride from the VLDL and give it to the LDL. Now take a look at hepatic lipase. Hepatic lipase hydrolyzes the triglyceride out of an HDL or out of an LDL. Remember, we are in a state of hypertriglyceridemia now. But these triglycerides in the VLDLs are not allowed to accumulate indefinitely. What happens is CETP and hepatic lipase go to work to correct this problem. CETP takes the triglyceride out of the VLDL and gives them to the LDLs in exchange for a cholesterol ester. So the cholesterol content of the LDL goes down, but the triglyceride content of the LDL goes up. Remember, the cholesterol ester and the triglyceride are the cargo, and LDL is the truck. With the LDL now rich in triglyceride, it becomes an attractive target for hepatic lipase in the liver. Take a look at this slide. Hepatic lipase then hydrolyzes the triglyceride in the LDL and removes that cargo from the truck. What you end up with is an LDL particle that is poor in cholesterol and also poor in triglyceride. Let's walk through this one more time. Hypertriglyceridemia means high triglyceride in the VLDLs. With lipoprotein lipase turned off, and the liver continuing to package the excess triglyceride into more and more VLDLs, you end up in this state. The triglycerides are not allowed to accumulate indefinitely. So CETP takes the triglyceride out of the VLDL and puts them into the LDL in exchange for cholesterol ester out of the LDL. This results in a triglyceride-rich and cholesterol-poor LDL. This type of LDL is super attractive to hepatic lipase and ends up having its triglyceride hydrolyzed by this enzyme at the liver. This results in a smaller and smaller LDL particle. When the LDL particle changes its shape, because its contents have now been removed, the ApoB on the surface gets altered and essentially deformed. Now you would think that this empty LDL would just get removed from the bloodstream by the liver because it really has no purpose anymore. It has no cargo to deliver. But because the ApoB on its surface gets deformed, it can no longer be recognized by the LDL receptor on the liver, and therefore it remains in the blood. And this doesn't just happen to one LDL, it happens to lots of them. So these small, obscured LDL particles begin to add up in the bloodstream, increasing their count and concentration. Remember, to wreak havoc in the blood vessel wall, the particle needs to be small enough to fit between the endothelial cells. Additionally, remember that the more of these small particles that are in the blood, the greater the concentration gradient, therefore driving force to essentially push those small particles deep into the subendothelial space. Once again, in a basic blood lipid panel, the cholesterol content of the LDL is measured. This is what LDL-C represents. In this instance, the cholesterol has been taken out of the LDL, so a basic blood lipid panel will show a normal or low LDL cholesterol. This can mislead some interpreters of the test, and the risk of this individual will be missed. Back to the atherogenic lipid triad. We understand now how hypertriglyceridemia develops. We also know how LDL particles become small and accumulate in number in the bloodstream. But before we discuss how HDL is affected, let me reiterate a few additional points about this small LDL particle. This small LDL particle now has a funky and deformed ApoB on its surface. This deformed ApoB prevents it from being cleared by the liver and additionally makes it more susceptible to binding to the proteoglycans in the arterial wall. Basically what this means is the funky ApoB causes it to get stuck in the endothelium. And this nasty little deformed ApoB is easier to oxidize. So if the particle can easily enter the endothelium, easily get retained in the endothelium, and now easily be oxidized to cause macrophage scavenging and the formation of a foam cell, we've basically started atherosclerosis. All right, on to the HDL. Remember, in this hypertriglyceridemic state, the VLDL particles are overly packed with triglyceride. CETP can also do a second thing to fix this situation. If you remember from the enzymes slide, CETP can also exchange one triglyceride for one cholesterol ester between the VLDL and an HDL. So this means that a triglyceride is taken out of the VLDL and given to the HDL at the same time a cholesterol ester is taken out of the HDL and put into the VLDL. This results in a similar particle composition as the LDL from earlier. The HDL ends up being triglyceride rich and cholesterol poor. This triglyceride-rich HDL is also very attractive to hepatic lipase on the liver cell. So once again, hepatic lipase hydrolyzes the triglyceride out of the HDL. But before we talk about what results of this, let's do a quick lesson on how HDLs are formed. In a normal insulin state, the chylomicrons produced in the intestines carry with it the apolipoprotein called ApoA1. This is the apolipoprotein that is found on the HDLs. This is one way an HDL starts. Lipoprotein lipase zaps this chylomicron and allows it to release the ApoA1 out into the bloodstream. Then, the adipocytes, which possess multiple transport proteins on its surface, expresses the cholesterol ester out through these transporters and further lipidates the ApoA1. Basically, the adipocyte dumps cholesterol ester onto the ApoA1 and grows a baby HDL. This HDL can be lipidated by other means as well to grow bigger and bigger. But this is what happens during normal insulin circumstances. As insulin resistance develops, once again, things get wonky. Insulin resistance renders LPL inactive, which causes the chylomicrons to release very little ApoA1s into the bloodstream. Additionally, insulin resistance reduces the expression of the transport proteins on the adipocyte, and thus HDL biogenesis is reduced. Now, back to hepatic lipase. This HDL that is subjected to hepatic lipase on the liver is the HDL that is present during insulin resistance. It is so depleted of its contents that once it comes in contact with hepatic lipase, the whole particle catabolizes and essentially disappears. Because of all of these abnormalities due to insulin resistance, HDL-C in a basic blood lipid panel will present as low, meaning worse. Back to the atherogenic lipid triad. We have the presence of hypertriglyceridemia. We have numerous tiny little LDL particles, and now we have low HDL. You can see how these three components are mechanistically connected and are significant promoters of the atherogenic cascade due to the end result of these small LDL particles. In a basic blood lipid panel, we can evaluate two of these components. But what measures can we ask for at the doctor's office to know our LDL particle size and count, and therefore truly understand where we stand when it comes to atherogenic risk? Let's begin with a simple blood test called ApoB. This blood test is a protein assay that pretty much any basic blood lab can run. It doesn't require specialized machinery, it just requires a simple kit. Every chylomicron, VLDL, IDL, and LDL each have one ApoB on their particle. So if we can count the number of ApoBs, we can count the number of these particles. 98% of all of these ApoB particles are LDLs. So this test is primarily counting our LDL particles. Two other tests exist that can measure particle size or density of each individual subclass. Nanomagnetic resonance spectroscopy, or NMR, is one of these tests, and electrophoresis is the other. These tests essentially measure the size of the magnetic field, or the size of the electrical charge field, that each particle emits, and can therefore estimate the size of the particle. Very specialized machinery is needed for these tests. Advanced medical and research labs may have these machines, but typically they are not present in rural hospitals like ours in Gunnison. Doctors, however, can ship your blood samples to these advanced labs. It just might cost a bit more for the test. Lastly, the triglyceride-HDL ratio can actually be calculated from a basic blood lipid panel and therefore inform you if it's necessary to ask for additional advanced testing. Remember, ideal is a low triglyceride number and a high HDL number. This would make the ratio low. If the opposite is occurring, such as in the atherogenic lipid triad, the ratio number will be high. The closer to 1 the ratio ends up, the better. If the ratio ends up greater than 3, this is a sign of greater risk for atherosclerosis. The ratio number is positively correlated with the number of small LDL particles. The higher the ratio, the higher the small LDL count. As we've learned, insulin resistance is a significant foundation for lipid abnormalities. So you could ask your doctor also for more advanced glucose or insulin tests, such as fasting insulin and fasting glucose, or the homeostatic model assessment of insulin resistance for glucose regulation. Well, that wraps up this lecture on the atherogenic lipid triad. In conclusion, we know that the lipoprotein particle is the carrier for our lipids that move around the body. We also know that insulin resistance disrupts the normal function of this lipid trafficking. And when things get wonky, hypertriglyceridemia, low HDL-C, and small LDL particles can result. And this is a recipe for atherogenesis. A simple way to begin exploring our risk is to calculate the triglyceride-HDL ratio from a basic blood lipid panel. From here, you can determine if more advanced lipid metrics are worthy. Thank you everyone for tuning in with me on this lecture. If you have any questions at all, please don't hesitate to email me at lesmith@western.edu. Also, all papers used as resources for this lecture are available upon request. Just reach out if you'd like me to send you some sources. Thank you for your time.